And yes, we do have a lot to cover today and a lot of details on inflammation. And we're going to start with the complement, but I'm not going to be able to finish everything in the complement because it's like way too much stuff and it's quite important, right? So I don't like to just dump stuff on you and say, you know, you're responsible for all this, especially with immunology. I think it's kind of like, well, let's see how much you can cover, and I will be, you know, I am as fair as I can possibly be. So yes, exam is on Tuesday, next class, right? And I will include this PowerPoint for the exam, right? But what I had planned to also do, like, you know, finish complement and do antibodies, that's going to be impossible. It's just way too much. Okay, so I'm going to cover the things here for this PowerPoint, and whatever we didn't cover that I had planned for this week, then it gets pushed to the next unit exam. Everybody cool? Okay. All right. So we had seen before the oxidative burst, right? We have seen the phagocytosis and starting usage of the lysosomal enzymes. We have seen the the enzymes that work at neutral pH and the azurophilic enzymes that work at more acidic pH, right? And then we went through a lot of details on the mechanism for oxidative burst. So I put this slide here just to review with you what is it that you need for oxidative burst, right? You are going to need molecular oxygen. You are going to need an electron carrier because you're going to put electron on molecular oxygen. Remember here, your electron carrier is an ADPH. And the enzyme that you use for transferring electron to the oxygen is your NADPH oxidase, okay, NADPH oxidase, right? And you are going to get several products out of this, but I had mentioned here superoxide and hydrogen peroxide, right? So superoxide is highly reactive. So in order to not have superoxide flying around the cell and destroying everything, you have an enzyme that transforms that into hydrogen peroxide. And the name of that enzyme is superoxide dismutase. It dismutates the superoxide. All right. One thing that I didn't mention before is that when you produce hydrogen peroxide, well, that is going to kill a lot of bacteria that are susceptible to this kind of killing. But you have certain bacteria that don't really care much about hydrogen peroxide because the bacteria, they have an enzyme that breaks down hydrogen peroxide into what? Water. And what is the name of the enzyme? Something? Catalase, right? The catalase. Catalase enzyme breaks down hydrogen peroxide to water, okay? So bacteria that have catalase or catalase positive bacteria, they can actually escape this type of killing, okay? All right. So in the meantime, with this oxidative birth pass, you also produce nitric oxide, and this is quite important because you get, you know, together with the reactive oxygen species, but nitric oxide is going to be important also to kill pathogens and as a vasodilator. Like, so nitric oxide is very, very potent vasodilator. Okay. So we have seen the chart of strong activators, the PRRs, that are strong activators for this oxidative burst, right? And I forgot to mention this guy over here, okay? This is called F-methionine and some other residues. And this is a receptor, okay? So this thing is LF receptor. What this thing is a G protein, it's a G-couple receptor protein on the surface of the cell, right, of those phagocytic cells. And what it does is that it's a sensor for this F-methionine that is present in proteins of bacteria, right? But is not present in proteins that are eukaryotic, from eukaryotic cells, okay? So F-methionine, you remember what methionine is when you're making proteins? The start amino acid for formation of proteins, right? So since you have a bacteria that has a different amino acid that starts the proteins, it's a good idea for your phagocytic cells to have a sensor to detect that very basic difference between proteins that are from bacteria. Okay, so this G protein couple receptor also activates this respiratory burst. Okay, besides that, you also have this other receptor that we're going to see a little bit more about it today, that is called the CR1 receptor. So this is a receptor on the surface of your phagocyte, but it's going to bind to a protein that gets cleaved out of your complement system. Okay, so that protein is called C5a. So what is this going to do? C5a is from your own complement system, binds to CR1, but it's not just going to bind to it and activate it. It's only going to bind to that if it is attached to a pathogen, if it is close to a pathogen that has been attacked by the complement system. Okay, so C5a connecting to this complement receptor 1 also activates this oxidative burst. All right, so then we left off on this side talking about your NF-kappa B, right? Remember, NF-kappa B is a transcription factor that is ready to do its job, but it cannot do its job because it's held on the cytoplasm by that inhibitor, right? So what is holding this transcription factor is the inhibitor of kappa B, right? So inhibitor is sitting there and nothing is going to happen to it unless the cell gets a signal that NF kappa B should do its job, okay? So what is the signal here? You're actually going to need two signals, okay? So the first signal here is going to be a binding of your PRR to your PAMP. All right, so that's going to send a signal transduction. Now this part here of this signal transduction is actually important because it is a cascade and it amplifies the signal received by the binding of the PRR. Okay, so this is an amplification cascade that is going to allow activation of a lot of your kinase, right? Inhibitor of kappa B kinase. So this kinase is going to go to kappa B, is going to put phosphates in it, is going to dissociate inhibitor kappa B from your kappa B, and then this transcription factor can go and do its job. All right, so that should be cool. Everybody's cool with that? All right, so what happens after you activate NF-kappa B? This is a very, very potent inflammatory process that needs to be controlled. If you activate too much of it, you can actually die from the consequences of too much activation. Okay, and this was actually one of the problems during the COVID-19 pandemic, because people who are always very healthy, had great immune system and never got sick, when they got infected with the COVID virus, they had too much of a reaction and too much inflammation and activation of NF-kappa B. Okay, so you end up with a lot of pro-inflammatory cytokines. And look at the consequences here. Okay, so here is showing you bacterial infection, but it could also be because of viral infection, right? So what is going to happen here? You're going to activate your monocytes, macrophages, right? You're going to have recruitment to the site of inflammation, but at the same time, you're going to have a whole thing that is happening with your endothelial cells. Okay, and this is more on the part of the inflammation that we're going to see now, right? So you have your endothelial cells. Something is going to happen here. Your permeability of your endothelial cells is going to increase. That means the barrier, your vascular barrier, is going to go down. If you increase the permeability of your blood vessels, means cells and liquids can come out of it. So if you have volume from your blood that is coming out through this permeability to the tissues, that means your total blood volume is going to go down. And that can cause shock, right? So in this case, because it is due to an infection, either bacterial or viral, that can cause septic shock. And can you die from septic shock? Yes, you can die from septic shock. Okay? All right. Something else is going to happen with your endothelial cells. You're going to increase the expression. So here endothelial cells, you're going to increase the expression of those little adhesion molecules. Okay, right there on the top of on the surface of those cells. So these adhesion molecules are going to interact with leukocytes that are floating in the blood, and it's going to attract the leukocytes to go to those areas and get out of the blood stream and go to peripheral areas. Okay. Besides that, you're going to activate platelets. Right, because platelets are very good at getting bacteria stuck to them. All right, you're going to start activating platelets. That also, what it can do is it can start activating a platelet plug, and that is part of coagulation. All right, so this would be just spontaneous coagulation that would be happening in case you have increased inflammation. Right. And we have seen a lot of this happening with the COVID infection, especially because COVID has a target receptor that is also part of endothelial cells. So if you have COVID on top of these cells and then you're activating platelets and you're forming a platelet plug and causing coagulation, that means you're going to spontaneously have blood clots, and those are called microthrombi. Okay, so it's right here, immunothrombosis. Okay, so you have formation of blood clots, and of course you can die because if you get a big blood clot that is traveling in your bloodstream and either blocks heart. Artery, or it goes to your brain and causes a stroke. Okay, so important things here that can happen. All right, so NF-κB is also important for activation of the complement system. Right, so you have molecules that are cytokines that are getting produced, and those are very good attractants of the complement system and increases production of complement system pathway. Okay. All right, so that's systemic side. All right, and that leads to the next what is the word? Strategy for killing bacteria, right? So let's say that your NF-κB activation wasn't really enough to get rid of the pathogen. So now you're going to activate another cellular pathway that is going to cause even more inflammation and is going to cause death of the cell. It's going to like burst the cell, kill the cell. Okay. So this is the example we have for the NLRP3 inflammasome activation. Right. So you can think of this as formation of little scissors inside of the cell. They're going to be getting together. You're going to have this aggregation of proteins, and this scissor is just going to punch holes in the cell, like from the inside out. You know, like if you watch those horror movies with aliens, and you get the alien like coming out of the stomach of, you know what I'm talking about? Yeah, so that's the NLRP3 inflammasome kind of idea. Okay, it's gonna kill the cell. All right. But you're not going to activate this inflammasome just by NF-kappa B activation. Okay, remember, NF-kappa B is the transcription factor, right? So after activation of that transcription factor, you're going to need a second signal from the cell saying that you need to kill the cell. You're going to need a stress signal, okay? So this is when the second signal comes along, all right? So this is going to be some kind of intracellular signal that is really going to trigger this inflammasome assembly. Okay, so what kind of signals do you have? Well, you can have potassium leaking out of the cell, right? Because remember, because of the sodium-potassium pump, you get potassium inside, sodium outside. In this case, you're going to have leakage of potassium. And that could be due to internal stress, but it could also be due to certain bacterial toxins that can punch holes on the cell membranes of the host, right? And once the holes are there, then the potassium can come out of the cell, right? So potassium efflux go hand in hand with membrane damage, right? This is stress signal. But you could also have lysosomal damage. Remember, lysosomes have those enzymes, right? It's the stomach of the cell, right? Now, what can cause lysosomal damage? One thing is DAM, that is uric acid. That's the thing that causes gout, right? And NLRP3 inflammasome is the mechanism of gout, okay? So uric acid can punch holes in the lysosomes and let all of those enzymes to leak into the cytoplasm, and that's a very stressful signal. With the same principle, you can have asbestos or silica or even cholesterol crystals that can punch holes in the lysosomes and cause this stress signal, okay? So now you should be thinking, what do you mean cholesterol signals? So if you have high cholesterol, what does that really mean? So this is when people started thinking, it's better to keep cholesterol levels down, to keep inflammation down. So let's give you statins. Great idea. No, not a great idea. Okay, this has a whole mechanism how that works, right? The cholesterol is not really the enemy. The cholesterol is actually a defense mechanism to get rid of the initial insult to your blood vessels, right? Cholesterol says causes atherosclerosis, and then you have these immune cells that go there, gobble up the cholesterol, and you get inflammation and all this stuff. No. What is the main cause of the initial irritation that would lead to atherosclerosis? Everybody awake? I can hear the clock. I changed the battery on that thing. Sugar, all right, it's sugar. Too much sugar in your bloodstream, bad idea. You're going to activate inflammation. So sugar in the diet will cause inflammation, and this is how, all right? Okay, so lysosomal damage, big problem, stress signal. You have other stress signals that can come from mitochondria that are stressed out. If they make too much reactive oxygen species, or if the mitochondria decides that it's time for the cell to die, mitochondria is going to start pushing out cytochrome C, or even its own DNA, right? And DNA of mitochondria is not supposed to be floating around in the cytoplasm, it's supposed to be kept inside of the mitochondria. What happens if the DNA of the mitochondria floats around in the cytoplasm? Is going to be detected by some PRRs, right? Because remember that symbiotic relationship of mitochondria, how we got mitochondria in the first place, right? Well, but we do have PRRs to detect DNA that is not supposed to be in the cytoplasm, right? So that's another stress signal that can cause this second activation and NLRP3 cascade. All right, so what is happening here? You have your initial activation, PAMP or DAMP. you have your NLRP3 that is in the cytoplasm, minding its own business, is not doing anything. But after you get this initial activation, then something is going to happen, right? Ultimately, you are going to get NLRP3 together with a binding molecule that I don't care if you remember that or not, because I never do. So you get a binding molecule, and you are going to get this other guy here that is a pro version of the final protein. So this one is not active. So this guy is a pro-caspase 1. If you cleave pro-caspase 1 and you make caspase 1 out of it, that's where, when you are going to trigger a couple, in this case, 2 forms of cell death. One is apoptosis. So caspase 1 is a apoptotic molecule. Okay? Once it's triggered, that's it. The cell's going to die. Okay? And the other consequence of this is that you are going to cause the production of inflammatory cytokines that are ready to be activated. Okay, so you have the pro version of inflammatory cytokines that are getting produced. This came from your NF-kappa B activation. You have the pro-inflammatory cytokines that are still not active, but once you get your caspase 1 activated, you are going to create the active form of those inflammatory cytokines. Okay, so you're going to cleave the pro version, and you're going to have exocytosis or secretion of your inflammatory cytokines. There are several inflammatory cytokines that are very potent, and I had mentioned before interleukin-1 beta, right? And here is another one, is interleukin-18. Okay, so these are 2 important inflammatory cytokines that are coming out of this pathway. Okay, so the formation of this NLRP3 complex, okay, is going to cause not dimerization but polymerization of proteins here. They are going to extend and extend and extend and cleave also inactive proteins to active forms that are going to attach to the cell membrane and punch holes in the cell membrane. Okay, so this form of death by activation of pore proteins by the inflammasome, this type of cell death is called pyroptosis. Okay, and this is inflammatory. So this is a kind of cell death that is damaging the cell and is causing inflammation. Okay, so you call that pyroptosis. It's different from necrosis because, like in necrosis, you have an external agent or something that smashes the cell, breaks the cell, and then you have inflammation that also happens. So this is coming from the inside, okay, from this process. And is also different from apoptosis. Remember, caspase 1 causes apoptosis. In case of apoptosis, you have non-inflammatory cell death. That's programmed cell death that is not inflammatory, right? And it's important to keep in mind there's different types of cell death, all right? So this, the pyroptosis as well as necrosis, those are highly inflammatory when you compare to apoptosis that is not inflammatory cell death. Okay, so what is happening over here? So this is when I played that little video. Hopefully it's gonna work.
Inside a body cell, there are danger-sensing proteins called inflammasome sensors. These dormant proteins detect when cells become damaged or infected with microbes. When
Can you hear that?
signals are present, these proteins are activated, and the proteins begin to form an inflammasome.
All right, so that's the inflammasome formation. Okay, see how it polymerizes.
Inflammasome is assembled. It activates a type of molecular scissor.
So it serves as an enzyme.
Other dormant proteins into their active forms. One
Okay.
dormant protein activated by trimming punches holes in the cell's membrane. Other proteins activated by trimming are released by the cell to recruit inflammatory cells and drive inflammation. Fluid flows through the holes into the cell, causing the cell to burst. Many microbes can't replicate when a cell bursts, and more inflammatory mediators spill out into the affected tissue. This cell death pathway is called pyroptosis. Inflammatory mediators released by the dying cell attract immune cells from blood vessels. These cells include phagocytes, who are
There you have your
cleaning
chemotaxis.
up all the debris, microbes, and dead cells, so that tissue repair can begin. The inflammasome pathway usually restores our health, but when this pathway keeps firing, it can cause a myriad of diseases, such as gout, chronic liver disease, and neurodegenerative diseases.
Okay, so that was a little movie on the inflammasome. That was the best one that I found, actually. This guy over here, okay? So how are you going to get NLRP3? Well, in case you have, again, signaling through PRRs and NF-kappa B activation, right? And having those stress signals, right? But you could also have other backup inflammasomes in case, you know, this one did not pick up. But it's basically the same type of mechanism. And I'm just, you know, going to focus on this guy over here because you have a lot to remember already. So what's going to do? You're going to activate caspase 1, and you're going to get cleaved forms of your interleukin-1, beta, and interleukin-18. All right. Okay, so this is the other path here that you have of innate system that is very important for killing of viruses. So that's the path that is the IRF, or interferon regulating pathway, interferon regulating factor. So here there are actually two pathways that will converge and give you the same type of response. So the two pathways are like interferon IRF3 or IRF7. The main idea is that you have your TLRs, and what kind of TLRs are those over here? They are on the membrane, or these are in the endosome, okay, in the endosome, and they are detecting some RNA in viruses. So you know that when you activate TLRs that are detecting viral RNA, you're going to have to upregulate antiviral response. So this is what this pathway is telling you, because interferon is really the first line of defense against viruses, right? So you activate, you dimerize here your PRRs, you send a signal to the nucleus that has to do with this IRF. IRF gets there, and is going to produce interferon type 1, which is alpha beta. Okay? When you produce this interferon, you are going to have certain genes that are antiviral that are going to get upregulated. Okay? Besides those genes, you can also activate your macrophages, right? You're going to activate the dendritic cells. Look at this, you're going to activate your natural killer cells, all right? And besides all of this, you're going to start increasing the expression of that MHC1 molecule. And that guy is what's going to present peptides to T cells. So MHC1 is going to activate what type of T cell? Cytotoxic T cell. MHC1 goes with CD8. 1 times 8 is 8. What is the MHC that activates CD4? MHC2. All right, so these guys here are going to activate the killer cells, the cytotoxic T cells. Okay? And they're also activating natural killer cells. Okay? So this is a type of a pathway that is really going to go after virus-infected cells. Okay. And remember that you have redundance on the functions of those PRRs, right? You have PRRs that can detect the same thing. You have a certain PRR on the surface of the cell that is going to detect viral RNA. In case the virus infects the cell and gets inside with the RNA inside of the cell, you can detect in the cytoplasm the viral RNA by cytoplasmic PRRs, right? So the example here of detecting cytoplasmic viral RNA is RIG-I, okay, RIG-I and MD5, but I usually remember the RIG-I. So reason for the redundancy, if the virus escapes the first detection, we'll catch him with the following detection. Okay. Something like interleukin 13, it causes some reactions in the intestine that increases peristalsis. And what does that mean? Diarrhea, big time, diarrhea. Bowel movement, you can cause movement naturally without going through the pain of those cytokines. But when you activate this type 2 response, you're going to cause very, very strong peristalsis. Okay, so you can imagine, not fun, all right? So that's type 2. And with these guys over here, you are going to cause a type 3 response that is inflammatory response. So these guys here are extracellular. Okay? They are extracellular bacteria and fungi. That is going to cause a lot of inflammation. So you should look here, what are the cytokines that are important, that are very inflammatory? Look at that. TNF alpha, interleukin 6, interleukin 1, alpha beta, interleukin 18, right? That soup of cytokines is highly inflammatory. And 17 as well, so 17 is a common one for both of them. Okay. All right, so now looking a bit more at the inflammation, right? Really got to be pros at inflammation. What is happening, what is causing, how is it going, how the cells are getting there, okay? So just in case you have some time left and you're still playing your immune quest, so that's what you're going to find in the immune quest, right? You're going to find extravasation of those leukocytes at the site of infection, and you're going to find activation of your complement system, right? We're going to get there. We're going to get there. All right, so first we get to look at inflammation. When we talk inflammation, we're really talking about acute phase inflammation. Starts quickly, resolves quickly, all right? When do you have a type of acute inflammation? Well, you must have breached your your basic immune defense, your level 1, right? So now you have penetration or intrusion of pathogen from, let's say, from your skin. Right? And I'll give the example, like when you go to a manicure, right? Not my example, but some of you go to a manicure and you have like the, you know, those cuticles, guys, that take like those excess skin out. You know what I'm talking about? A girl-like thing, you know, usually, but some guys do manicure too, which is just fine. I never had time, but anyway, sometimes you get a bit of inflammation, and you know, if they nip on the skin, and that thing hurts, right? It hurts really. That's why I don't like manicure. It's not because I'm not too girl-like or I don't have the time. It's because of the inflammation. Yeah. Okay. So what happens? You have a lot of pain, right? You have swelling, right? Swelling. You have redness, and the thing is hot, right? So that is acute inflammation. Okay. So what is happening here? Because of either damage or bacterial infection, you are going to have local upregulation of those inflammatory cytokines, and those cytokines, they're going to get into your bloodstream, they're going to travel everywhere in the body, they're going to get to the liver. Here is the liver, okay? So here are your cytokines getting in the liver, and in the liver, it's going to start activating the production of acute phase proteins, all right? So here are some proteins you should keep in mind. C-reactive protein, and this is a big one, complement protein 3. C3 is the most important complement protein. And you're also going to have this fibrinogen. What is fibrinogen good for? Blood clotting, right? That's the second stage of blood clot. First you have your platelet plug, and then you have your fiber formation. You have your clot formation. So this is the coagulation part. Fibrinogen is important for that. Okay. All right. So how much are these proteins increasing? A lot. Look at this. Up to 1,000-fold of those acute phase proteins. So it's a lot of upregulation of these proteins. Okay. All right. So this is just for you to study, and I have just mentioned that. Remember here, your C-reactive protein also serves as a PRR, right? Okay. So here, what is happening with inflammation? First, what is going to cause inflammation? Of course, you're going to have infection. You can have tissue damage. That's the case with the, you know, with the manicure, the nipped on your finger, and that's damage of the tissue. You always get some bacteria that gets inserted with that, right? Or you can have some toxins, like some acids. Acids burn, so that will cause inflammation, right? Foreign substances that get inserted into the body, that could also cause inflammation. Right? That would be like piercing, for example. All right, so what are you going to see? You're going to have a lot of heat. It's going to get red. You're going to have swelling of the tissue. And yeah, you're going to have a lot of pain. I like how they have this screw going through the brain. Painful. And this is the fifth sign of inflammation that we usually don't think about. That is a temporary, hopefully, temporary loss of function, right? So imagine if you have your finger that is really hurting and you have to do something, you go like this and you keep that finger away, right? Because it hurts, you don't want to touch it. It's a temporary loss of function. Okay. So we are talking about your acute inflammation. Characteristics of this is that it starts really, really fast, but then it resolves really, really fast. The main cell that governs this inflammation type is your neutrophil. Okay? So remember, neutrophil is has the highest percentage of white blood cells in the blood, and is always floating around, going everywhere. And when it finds an area of inflammation, it's going to go there, it's going to come out of the blood, to go through the blood vessel, and attack the problem of the infection or cause further inflammatory signals there. Okay, so neutrophil. When you compare this with chronic inflammation, then the situation is different. Okay, so first, what's happening with acute inflammation? These cells get ramped up, They do their job, eliminate the pathogen, that's really, really important, eliminate the pathogen, and then the inflammation resolves. Okay. With chronic inflammation, you don't eliminate the pathogen, or you don't eliminate the trigger signal. Trigger signal is always there. Okay, so what happens with chronic inflammation? It starts slow, but then over time it keeps on getting worse and worse and worse and worse, and it never stops. It's only going to get worse and worse until when? Very much. Until you die. Maybe not from the inflammation, but from something else. Nobody lives forever, right? Now, the cells that are most characteristic of chronic inflammation, those are the T cells. All right, so T cells are very important for chronic inflammation. Also the macrophages, but mainly T cells. Right? So now you know the difference between chronic and acute inflammation. I love to ask questions about that. Okay? All right. So here, when you talk about acute inflammation, you're talking about a normal process and is short-lived, and after that is going to help with healing of the tissue, and the thing's going to go away. So when you're talking chronic inflammation, you're talking about pathology. So it's going to stay and cause tissue damage, and really is going to be progressing throughout life, and it only gets worse. Is there something you can do about chronic inflammation? That was a simple question. Yeah, you can take some drugs. You always have a drug for something. All right. So yeah, you have some drugs that control the progress of chronic inflammation. You will not be able to stop it or reverse it, but you can slow it down a lot. All right. So here, cells that are involved in acute inflammation, as I mentioned, the most important one here is your neutrophil, and the rest for you to study. And molecules that are important for acute inflammation, right? You have here molecules that are going to dilate your blood vessels, molecules that are going to bring other leukocytes to the area, those chemoattractants, and same molecules that can dilate blood vessels, a lot of times they are going to increase the permeability of those blood vessels and cause some expression of molecules on the blood vessel cells. All right, so here are the molecules that you should absolutely be familiar with. Histamine, prostaglandins, and of course cytokines, inflammatory cytokines. All right. What's going to happen? Remember, you're going to have inflammation, pro-inflammatory cytokines. You are going to get the liver to produce those acute phase proteins. Your blood vessels are going to increase expression of adhesion molecules. Complement system is proteins are going to go up, right? And now we're going to see what happens next, right? Important molecules of acute inflammation, so we just put them out here so you can follow and study when you're getting ready, right, for the test. Remember I mentioned complement activation, especially C3 protein, okay? And here, endothelial adhesion molecule, we're going to see which ones are important, but this guy here, ICAM, is really an important one. Okay, it's called integrin. This is ICAM. All right. Okay. So here. All right. So let's see what is happening here with the inflammation, right? So this is an example of that cuticle thing that breaks through the skin. Right? So here you have damage, and you have penetration of those bacteria. Okay. When the bacteria come here to the dermis, epidermis is dead stuff mainly. Dermis, you have your blood vessels. And here is where you are going to start signaling white blood cell recruitment. Okay? You are going to start getting swelling. So blood vessels are going to get leaky. They're going to get more permeable. and you're going to have production of those cytokines and those inflammatory molecules like histamine, prostaglandins, right? And besides all that, you're also going to start having formation of blood clots. Because of course, you break your skin, you start to bleed. You don't want to bleed forever. So you want to stop the bleeding. So increasing blood clot formation is a good idea, right? Okay. All right, so here is when everybody starts panicking. So don't panic. The things should make sense, right? So what is happening with your blood vessels? So first thing that is happening, you are going to respond to the presence of those inflammatory cytokines, right? The blood vessel is responding, and this guy here, prostaglandin, bradykinin, leukotrienes, and also histamine right there, okay? Histamine and prostaglandin, those guys are very, very important for opening up your vessels and increasing the permeability. So you look at the blood vessel here, so this guy is like opened up, and between the cells, first you had very, very close proximity of the cells because you don't want anything to be leaking out of the blood vessel, but after histamine and prostaglandins act on the endothelial cells, you're going to have separation. You're going to have a bit of a window here that is going to allow fluids to come out of the blood vessel. So you're going to have plasma and proteins from the plasma, right? They are going to come out. So liquid and protein is going to come out there. But not red blood cells unless the blood vessel is actually damaged, right? So let's say this blood vessel is not really damaged. So you have increased diameter and increased permeability. Okay, so what is going to happen now? The same molecules that did that to the blood vessel, they are going to start increasing the production of those selectins. Those are receptors on the endothelial cells, receptors for something that is present in your white blood cells, especially in your neutrophils, okay? So increasing the selectins, and important one here is also, ICAM1, okay? Increasing selectin is going to allow interaction of your neutrophil to the side of the blood vessel. So E-selectin here is for endothelial selectin, okay? So let me see the next one here. Okay. So you also have the P-selectin. I forgot what the P is for, so of course I'm not going to ask you. But E-selectin is for the endothelial selectin. Okay. So now let's see what is happening here. You have blood flow going this way, right? Everything is going this way. In the middle of the blood vessel, you have the cells that are going really, really fast, right? So if it is going in the blood flow in the middle here, forget it, it's going to pass right by. And the analogy is like if you are driving on a highway, like you're driving like I-95, and you go on the left lane, that this would be in the middle here, right? Although this is only one direction, right? So you're going like in the fast lane, so you go really, really fast. If you find a site of inflammation on the side of the road, like, I don't know, you have a friend waiting for you over there, right? Calling you over there, and you are going really, really fast in the middle of the road, you're going to miss your friend, right? So you can see the idea here is like I'm driving and I see your friend, oh, hi, bye, and you miss them. You cannot get to the site of the inflammation, right? So what happens here is that when you have upregulation of the selectins, you are going to start having slow interactions or bind and release interactions. So those E-selectins and P-selectins are going to bind to some molecules on the neutrophil, and kind of like a bit like Velcro, bind and release, bind and release. It's not too much of adherence, but what that does, it keeps the neutrophil rolling on the surface of the blood vessel. Okay, so it's like rolling, it can interact and roll some more and roll some more. But then when you have expression of this ICAM1, Now you're going to have a strong interaction, okay? ICAM1 interacts with this binding factor on your neutrophil that is called LFA1, okay? So LFA1 binding to ICAM1 gives a strong pull that is going to cause the emigration or extravasation of that white blood cell. All right, so this is also called diapedesis. Remember the guy, diapedesis? Yeah, okay. So what, how are you going to remember that? I know it's a lot of stuff, like the selecting, you're selecting, as like, yeah, selecting, I'm not really attached, I can detach, I'm just rolling. So here, let's say this whole board has the selectins, but between, like in the open areas, you have, close to the open areas, you're gonna have this ICAM1. So I'm the neutrophil, so I'm here, I was running, but now I'm not running in the middle anymore, so I'm interacting with the selectins. So what is the process that happens with the interaction of the selectins? Slows down, you get the rolling. So that's, the rolling. I'm rolling on selectins, and can you stop? Yes, I can. Now I go to the site of inflammation. I'm not in the blood stream anymore. I will find the bacteria and kill the bacteria in the background, but this is how it gets out, right? This is capture, interaction with rolling, and here you're gonna have ICAM interaction. You really get stuck to it and you go through extravasation, okay? Everybody got that? I'll ask a volunteer next time. Okay, so here, this is what I had just talked about. Okay, immigration diapedesis, here we go. I just mentioned that. And we talked about chemotaxis before. All right, so here, one thing I didn't mention here, okay? So here ICAM1, and you have this other molecule, the PCAM1, that is important for allowing the cell, guiding the cell to go through that between the cells. You call that paracellular route, like going between the cell to get to the site of inflammation. All right. So this diagram, I think I have shown before, right? Did I show this before? No? Oh, so here I introduce your diagram of rolling and diapedesis. There's a lot of stuff going on here. Like, for example, at the site of infection here and inflammation, you have this neutrophil that is capable of phagocytosis. You also have macrophage over there that can do phagocytosis, right? So here you have a blood vessel, and you have some rolling going on over here. And here you're going to start adhesion to ICAM and diapedesis, right? You have some, red blood cells that keep on floating. The red blood cells are not going to come out of the blood vessels. Okay. Oh, finally, a question. Here. Which event occurs first in acute inflammation? Okay, you can consult each other. Just think what is happening. Are you going to get cells somewhere if you don't have anything that is calling the cells over there? Does anyone need more time? No, you all answered? Okay, what is your answer? C. All right. So I got some people saying A. So A is telling you neutrophils and other immune cells begin to exit the circulation and move into the tissues. Why would the cells go there if you don't have pathogen or damage calling them? Right? So you need to have some damage or pathogens that get activated because of the, in order to call all this process to happen. Okay, so let's see what's going on here. At the site of inflammation, so let's say this is, I don't know, some mosquito bite or insect bite or something. You do have, yeah, actually my son is very allergic to mosquito bites, and oh my God, it was horrible. You got to be hospitalized. He was only 1.5. It was really, really bad. Okay, so here you have your redness and swelling and everything else going on with it. And what you should know is that the inflammation is going to resolve like relatively quickly. And what's going to happen is that those neutrophils, right, they get to the site of the damage. Those neutrophils are very good at causing inflammation, but they're very, very short-lived, especially if they're not in the blood, if they are in the tissues, right? So they're going to die quickly. Now, before they die, they are going to start producing anti-inflammatory molecules, all right? And together with other white blood cells, like with some macrophages, you're going to start producing these anti-inflammatory molecules, all right? So here you have examples of lipoxins, okay? And you are going to have death of the neutrophil, and you're going to have macrophages that are going to go there and start to clean up and induce healing, okay? All right, so here I also give an idea, because when you have an inflammation, it's painful, right? So you have a option of using certain drugs that can inhibit the expression or the release of some cytokines in order to at least slow down the inflammation and slow down the pain. So these NSAIDs are examples of those drugs. So those are called non-steroidal anti-inflammatory drugs. So in case of ibuprofen, for example, what you're going to do is that you're going to inhibit an enzyme. So here is your cyclooxygenase enzyme. And if you inhibit the enzyme, so you're not going to be producing prostaglandins, okay? So if you cut the enzyme, you're not going to have so much of the prostaglandin. You're going to slow down inflammation and also the pain. Okay. So here, limiting inflammation and pain. Yeah. Mm-hmm.
Basically, our CNS is but the outcome is the inflammation.
Then you never know how somebody is going to react to certain drugs. But usually with anti-inflammatory drugs, you also have those side effects that can exacerbate the problem. But I did not look into the exact mechanism of how histamine can be exacerbated by these drugs, but you do have allergic reaction to certain drugs that can cause exacerbation of all pro-inflammatory cytokines. Okay, but I have not heard of allergy against NSAIDs. But I'll have to look into it and see what kind of mechanism it is involved, because it would still inhibit cyclooxygenase, but then you have other pathways that would increase more inflammatory cytokines. So Oh, yeah, that. Yeah, that is bad. Yeah, I know somebody who cannot take oxycodone. Yeah, imagine. All right. All right, so here a comparison with chronic inflammation. Okay, so in the case of chronic inflammation, you can never get rid of the stimulus, right? When you compare it with acute inflammation, you get rid of the pathogen and now the tissue will heal. Now, with the chronic inflammation, you don't get rid of the pathogen and you have this constant stimulation of the system. And eventually, you are going to activate your T cells to continue on that chronic inflammatory path. Okay? All right, so molecules here that are in relevance to T cells and inflammation. You have your TNF alpha, you have interleukin 17, and usually you also have some macrophage continuous stimulation that are feeding on the continuous production of those inflammatory, long-term inflammatory cytokines. Okay. So the connection here that, what happens after your stimulus is gone in case of acute inflammation, you are not going to have this M1 phenotype of your macrophage anymore. You're going to shift to an anti-inflammatory type of macrophage, right? So when your stimulus is gone, what you're going to do, you're going to start to produce anti-inflammatory cytokines, okay? So an example of an anti-inflammatory cytokine is interleukin 10, okay? And you also have TGF beta, okay? So these are two anti-inflammatory cytokines, and those are going to produce tissue repair. They're going to promote tissue repair. How are you going to remember interleukin 10? Well, 10 fingers, you're going to stop something, you go like, okay, so stopping the inflammation, interleukin 10. TGF, thank God it's Friday, beta, okay, so that's, anti-inflammatory party-like mode. Yeah, hopefully that one helps, okay. So this is just a quick review of what the macrophage is doing. Remember, it has receptors for LPS, and these are the types of cytokines are produced, right? Inflammatory cytokines and antiviral cytokines. And this is how the classical inflammatory M1 macrophage phenotype is working, okay? Highly inflammatory, highly phagocytic, okay? So let me see here. Okay, this is just more review on those inflammations. So here, example of diseases related to chronic inflammation, and they will tend to just get worse with time. And it doesn't really resolve, but you do have drugs to slow down the inflammation. So here are some examples like rheumatoid arthritis, of course, atherosclerosis, pulmonary fibrosis, cancer, Alzheimer's disease. Alzheimer's is inflammatory, is neuroinflammatory disease. Right. Okay, so I just mentioned about your anti-inflammatory healing macrophage. So now finally we're going to get to the complement system, and it's actually fun. Okay, no way. All right. So complement system. So you have those little guys saying, "You are so strong. Oh, thanks for the compliments." It's not a compliment system; it's a complement system, right? Oh, you are brave. Lots of complement proteins going around all the time. These guys are always in the blood and in the lymph, right? And they are surveilling for pathogens like all the time. So I put here the pages in the book that you can find that information, right? So basically you have three pathways for this complement system, okay? But they all converge at a specific point, and they're going to form these holes on the surface of the pathogen that you call it a membrane attack complex. Okay, so what's going on here? It's really a system that is evolutionarily preserved. Okay, so most species will have it. Okay, and the organ that is producing these complement proteins, the organ is the liver. Okay, so the liver here is producing your complement proteins. And And those are soluble proteins in the blood. So it's in the blood, it's really everywhere, okay? What is happening here? When the proteins of the complement system get activated, well, first, when they're just floating around and doing the surveillance, you call it a capital letter, capital C for the protein. When it gets activated, those proteins are going to break into two pieces. So in case of this guy over here, is going to be a larger piece and a smaller piece. The larger piece is going to be a B fragment. The smaller piece is going to be the A fragment. So the A fragment keeps on floating in the blood. So A fragment goes away, okay? And the B fragment is going to attach to the surface of the cells. Okay, so the B fragment binds and the A fragment floats away, okay? Once you have cleavage of this zymogen protein, you're going to start triggering a cascade of events. So you cannot stop that cascade. So once you have the trigger, you're going to get the cascade, okay? It's going to be so fast, and you're going to have production of the membrane pore kind of thing that's going to put holes in the membrane and kill the pathogen. Okay. Where is it you're going to find your complement system? In the blood. You're going to find the lymph, and in the fluid that is bathing your organs, like your extracellular fluids. Okay? You're not going to find it in your intestines, which is actually a very, very good thing. Because as you're going to see with these complement proteins, they tend to just bind to cell membranes. If they bind to a cell membrane of your intestines and decide to punch holes in there, that's a bad idea. That means you're going to have a lot of your microbiome that are opportunistic pathogens. They're going to get into your system and kill you. So it's a bad idea to have complement in your intestines, in your mucosa tissue, okay? No complement in mucosal tissue, okay? All right, so this complement is called complement system because it is really complementing the innate and the immune system. So it's a continuous surveillance system that is really helping both of those systems, okay? And is mainly protein surveillance, okay? Protein surveillance mechanism. Complement system is very, very important in increasing acute phase inflammation. Right, so it has proteins that once they're cleaved, they're going to cause more inflammation. Okay, so let's see what is happening over here. The whole idea of the complement system: you have the zymogen protein that gets cleaved. The B part of the complement protein is going to get stuck on the surface, and if it is the surface of a bacteria, it's going to flag the bacteria for destruction. You call that opsonization. Okay, so you have to, here's our little bacterium, and here is C3b that binds to the surface of the bacterium and is going to flag it for destruction. Now, if you get a phagocyte here, like a macrophage here, that gets in there and sees the C3b, it has a receptor for that, and is going to bind to that and is going to eat the bacteria. Okay, so opsonization means bacteria is tasty now. Phagocytic cell is going to go and eat it. Okay. Right. So I mentioned C3. C3 protein is the most important protein of the complement system because all of the different pathways, they are going to converge in this C3 stage. Okay, so if you have somebody who cannot really produce C3, the liver of the person is not very capable of producing C3, and you're going to have a big problem because now this patient is going to be susceptible to bacterial infections because they don't have this constant innate surveillance. in their blood, in their lymph, in their system. Okay, so if you don't have C3, that's a big problem. All right, so 3 pathways of your complement system. One of them is called alternative, the other one is lectin, and the third one is the classical pathway. The classical pathway is the only pathway that involves a component of your adaptive system. You need antibodies to activate your classical pathway. The only time you're going to get antibodies is after your adaptive system B cells had been activated. Okay? So at first, forget about the classical system. It's not going to be surveilling anything, because if you didn't see the pathogen, you did not activate the adaptive system, right? So the first one that comes into action is actually your alternative pathway. Don't ask me why. Immunologists always like to make things more complicated. First one, always surveilling, is the alternative pathway. Okay? So alternative pathway, what happens is that you have your C3, right, your complement 3 that is floating around, and it doesn't even need anything to do the cleaving of the A and B parts. It can cleave itself just by the presence of some water. Okay, so it does some auto-cleaving, and then it becomes C3b and C3a, right? And then this guy, C3b, can bind to any membrane that is around. If it happens to be membrane in bacteria, great, it's going to tag the bacteria for phagocytosis. But if it binds on a membrane that is your own cell, that could be a problem. It could tag your cell to be destroyed, and that's not good. So you do have defense mechanisms that are going to stop complement from attacking your own cells. Keep that in the back of your mind, okay? But the alternative pathway is when C3 just cleaves itself, okay? What does it attack? It attacks surface of pathogens, okay? And you're going to start creating the formation of your membrane attack complex. Okay, so this is the first thing that comes into action. Then after this, you're going to have your lectin pathway. This is when you're going to have those PRRs that are involved here. Okay, so floating PRRs, soluble PRRs, like mannose binding lectin. Remember this guy here? It's part of the complement system too. Okay, mannose binding lectin is going to recognize those patterns on the surface of cell, and this is when you need those patterns to be at high concentration, right? You have a high frequency, because this mannose binding lectin is that little thing that looks like a spaceship. If it binds only one side, it's not going to activate. If it binds 2 sides, now it's going to activate, right? So mannose binding lectin can also get together and get activated and cleave C3. Okay, so this guy here cleaves C3 by itself. Lectin pathway binds to the surface of bacteria and activates C3 to cut. Okay, and then the last one that comes into action is your classical pathway, in which you're going to have mainly antibodies against specific stuff on the surface of your pathogen, and the antibodies are going to recruit the cleavage of your C3. Eventually, they're all going to activate cleavage of C3. And then what is C3 going to do? This is what's going to happen. C3 is going to cause a cascade that is going to recruit inflammatory cells. So C3b gets stuck to the membrane, C3a floats away, but this guy here is a very strong chemoattractant of inflammatory cells. So C3a is going to call on recruitment of your inflammatory cells. Okay? Besides that, remember, C3b is going to bind to the surface of your pathogen and is going to flag it. It's going to cause opsonization. And then Syn3A causing the phagocytes that are going to eat the stuff that is flagged. Okay? And lastly, you're going to form the membrane attack complex that is going to put holes on the surface of the pathogen. Okay? And here, overall result of this activation of C3 from either of the pathways is that your pathogen is going to die, and either by recruitment of the cells or by phagocytosis or by having holes punched on the membrane, it's going to get killed, hopefully, right? 3 times. Okay. All right. So here is what is happening with your overall idea of your complement pathway. Remember, classical pathway uses antibodies. Lectin pathway uses PRRs, like your mannose binding lectin. In this one, they have mannan, right? That's like bacteria or fungi, right? And here is your first one that is acting. That's your alternative pathway, in which C3 just cleaves itself. Ultimately, you have cleaved C3, and you're going to get opsonization and membrane attack complex formation, right? Okay. So here is a little idea of, again, what is happening with overall idea of your complement. You have C3 cleavage, okay, from the 3 pathways. C3A is going to cause inflammation. This is bringing chemotaxis, right? C3B is going to cause opsonization, and down the line you're going to activate the other C molecules, and you're going to cause increased inflammation, just like C3A causes inflammation. Ultimately, you punch hole on the membrane and you make the pathogen die, bleed to death. This is the idea of the hole of the membrane that gets recruited by other complement proteins, and this hole in the membrane is called your membrane attack complex. Okay, don't go yet. Last point here. All right. The alternative pathway. What is happening here with your alternative pathway? Okay, what's your answer? B.